In this video, I will summarize differences between mitosis, shown on the left, and meiosis, shown on the right. Mitosis and meiosis look very similar to each other for starters. You have two cells, both present, each with a nucleus and chromatin therein. As things start to occur, though, it looks a little bit different. Starting with prophase. In this case, in prophase and mitosis, the chromosomes have condensed and they're just hanging out. Whereas over here in meiosis, homologous chromosomes are actually hanging out next to each other and crossing over and exchanging chunks of chromosomes. Once they get lined up during metaphase, in mitosis, the chromosomes are lined up in a single line along the metaphase plate, whereas in contrast, during metaphase 1 of meiosis, the chromosomes are lined up in their pairs, each one on either side of the metaphase plate. That means that once anaphase occurs, whole chromosomes go in either direction in meiosis. But over here in mitosis, you have just half of the original duplicated chromosomes. So now unduplicated chromosomes that are moving in either direction. So 4 total going north and 4 going south, in comparison to just 2 going south here and 2 going north. Eventually, nuclear envelopes form around these. And by the time mitosis is done, we now have a cell that still has the same number of chromosomes as the original cell. Look, there's 4 chromosomes. 1, 2, 3, 4. 1, 2, 3, 4 over here. Yes, those chromosomes are unduplicated, but the number of chromosomes did not change over the course of mitosis. In contrast, in meiosis, now we have half the number of chromosomes we originally had. Here's just 1, 2 chromosomes. 1, 2 chromosomes. At this point, mitosis is done, but meiosis is not yet done. There's a whole nother round of division that occurs in meiosis. So in meiosis, this time these still duplicated chromosomes will go through prophase, and then in metaphase get lined up just like in mitosis, right on the metaphase plate. The sister chromatids are pulled apart until eventually 4 gametes are formed that are haploid because they have half the number of chromosomes, just 2. And at this point, those chromosomes are unduplicated. They've become separated from their sister chromatids, which are now in another cell. Furthermore, you can see that because of crossing over and independent assortment, the combination of chromosomes in this cell is not quite identical to in this cell. This one has these 2 little patches right here that have been crossed over. And they're quite different from the cells that are down here. So while mitosis creates 2 cells that are still diploid, that have the exact same chromosomes inside, in meiosis you develop 4 cells which all have different combinations of chromosomes inside, making them very good genetically diverse gametes.